Last lecture, we talked about the mechanics of the secretory pathway, how vesicles form, how they're addressed, know how to fuse with a target compartment, etc. Now we'll talk about what a package experiences, what it's like to be a part distributed by this traffic system, starting with how things enter the secretory pathway in the first place. Proteins are directed to the endoplasmic reticulum co-translationally, that is, as they are translated. If we take an mRNA and imagine ribosomes recruiting to it, and I'm going to draw them upside down from conventional appearance for convenience. As a protein gets extruded from the ribosome, if it happens that this N-terminal 20 or so amino acids folds up into a helix with some hydrophobic residues on the outside, then it's recognized by a signal recognition particle, which is an ancient part of the kit of life. And the signal recognition particle docks this nascent polypeptide with a transmembrane pore, a protein pore, through which it is conducted, in the case of a eukaryotic cell, into the endoplasmic reticulum. This machinery is something we share with prokaryotes. Okay, I drew my RNA wrong. It needs to come down here a little bit to meet the, oops, get rid of that. Because in order to make the rough endoplasmic reticulum, which is basically endoplasmic reticulum studded with ribosomes, these things have to be tucking their products directly into this channel. In fact, that little N-terminal bit is turned around in there, and this protein, as it grows in the ribosome, is fed through the channel. As it grows, it develops its secondary and then tertiary structure, whatever folded shape it's supposed to adopt. Until, at some point, the ribosome is done, it's going to fall off and go look for business elsewhere. Whoops. Last step here. Something typically snips off the signal sequence, that N-terminal piece, and it stays behind there as this protein goes and completes its maturation, whatever shape it is supposed to end up, in the endoplasmic reticulum. Now, there's a special case for things that have transmembrane domains, that is, for things like cell surface receptors and so on. At any rate, proteins are tucked into the endoplasmic reticulum co-translationally once this signal sequence at the N-terminus is recognized by the signal recognition particle and docks it with this aqueous pore, a protein channel. So what do proteins do in the endoplasmic reticulum? Well, chiefly they have to complete their folding, which is a non-trivial process for many proteins. The endoplasmic reticulum is full of chaperones that help proteins fold. One of them is called BIP, and Guess what BIP stands for? That's right, binding protein. No kidding, because it's a binding protein. It binds proteins as they fold and helps them to negotiate the complex process of finding the most stable folded configuration they can. Proteins that don't succeed in this are exported through another protein channel tagged for degradation and chewed up in the cytoplasm. So protein folding is accomplished in the ER, and once a sufficient number of proteins that are correctly folded, and usually marked with a sugar tag to say that they are so, once a sufficient number accumulates in the endoplasmic reticulum somewhere, then they will group together and vesicle formation will be initiated, if there are enough of them, for transport onto the Golgi apparatus. Proteins destined for secretion move stepwise from the endoplasmic reticulum to the Golgi apparatus. The port of entry is called the cis-Golgi network. Then there's the cis, medial, and trans-Golgi stacks, and there might be several more of them. And finally, the trans-Golgi network, where they're actually packaged into secretory vesicles and head off towards their final destination. What happens in the Golgi apparatus? Well, a number of things, but one is that proteins are decorated with a succession of covalent modifications. Proteins might find other members of their mature complex in the Golgi and get together, etc. There are several other destinations for vesicles from the Golgi apparatus again, and so the Golgi apparatus also sorts proteins to endosomes, or sorts them to the regulated versus the constitutive secretory pathway, for example. So since by default proteins that enter the ER are heading on to the Golgi, how do proteins that reside in the ER stay there? Well, for one thing, they may not have the signals that stimulate vesicle formation to get them shipped off to the Golgi apparatus, but sometimes surely by accident it happens, that you consider some protein that's supposed to be in the ER, something like BIP, for example. Now my puzzle piece is going to be an ER resident protein. Surely by accident, once in a while, it gets into a transport vesicle. Now this is supposed to be the cis-Golgi network. Within the cis-Golgi network is a protein that recognizes proteins that are supposed to stay in the endoplasmic reticulum. This is called the KDEL receptor because the amino acid sequence lysine, aspartate, glutamate, leucine tags proteins for retention in the ER. If you stick this little sequence on the C terminus of some protein that's otherwise destined for secretion, you can force it to remain in the endoplasmic reticulum. The KDEL receptor's job is to go and catch little puzzle pieces that make it in here by mistake. It gathers them up and stimulates vesicle formation around them. And I forget what color they were before, but of course the KDEL receptor is going to have to be recruiting COP2 coats to make vesicles headed back for the endoplasmic reticulum. So this recycling pathway between the endoplasmic reticulum and the cis-Golgi network is particularly well understood, but there must be recycling pathways amongst many of these membranes. There is also a well understood one between endosomes and the Golgi apparatus, mediated by the retromer proteins, for example. This brings up a puzzle of its own that illustrates a very important dynamical principle about the operation of the endomembrane system, and it goes something like this. Let's schematically redraw this in simplified form so that here's the endoplasmic reticulum, and it's loaded with some ER resident proteins that it wants to hang on to. Apparently, it can't help once in a while getting them into vesicles for transport out, perhaps just passively, that is, they diffuse in there and they get transported to the Golgi apparatus, where they don't belong. Of course, if all the ER resident proteins did accumulate in the Golgi apparatus, in the cis-Golgi up here, then, well, the cis-Golgi wouldn't exactly be the cis-Golgi anymore, would it? It would be the endoplasmic reticulum. So that's the role for the KDEL receptor and other retrieval pathways, is to gather these things up and ship them back. But look at what we're asking the KDEL receptor to do here. So if the KDEL receptor is sitting in the Golgi waiting to gather up these proteins to be retrieved and bring them back, then the KDEL receptor must surely spend some time here in the ER. It must surely itself be getting shipped out to the Golgi, in fact. There's got to be plenty of it in there. We're asking this receptor to bind its ligands, these guys, at low concentration, but release them at high concentration. How is this retrieval process supposed to work if these two compartments differ in the opposite sense from expectation for the simple kinetics of receptor-ligand interactions? The answer is the pH gradient within the endomembrane system. When I first drew this, I said that the purple here, the mitochondrion, represents, pH 8, right? That's higher than the cytoplasm. The endoplasmic reticulum, the internal pH is probably around 7. That's maybe a little more. That's not too different from the cytoplasm. Whereas at the opposite extreme, down in the lysosome, the pH is something like 4.5 or so. And in the Golgi apparatus, the pH might be 6 or 6.5, depending on what part. In fact, the pH declines successively over the progress through the secretory pathway. So that's the trick. It turns out that the KDEL receptor's affinity for the sequence, which you'll notice consists of 3 ionizing residues. The lysine is basic, and the aspartate and glutamate are acidic residues. The affinity as a function of pH from the pH in the Golgi to whatever it is, higher, looks like this. That is, at the pH typical of the ER, the KDEL receptor has low affinity for its target sequence. At the pH typical for the Golgi, it has high affinity. This pH gradient, again, operates throughout the secretory pathway, and one of the important consequences that I want to highlight for a moment before we move on is that whatever is getting manufactured in the Golgi apparatus must tolerate relatively low pH. And as a preview of a different topic, that must include mineralized objects that are made within the Golgi apparatus. The calcium carbonate precipitation on coccolith scales, for example, or on the spicules that make up echinoderm skeletons, all that takes place within highly expanded compartments of the Golgi apparatus, takes place within the endomembrane system. And so despite what you may have heard about how ocean acidification threatens to prevent organisms from calcifying their shells properly, if they are making them within the endomembrane system, it must be that that process is intrinsically tolerant of relatively low pH, because this pH gradient organizing the secretory apparatus is, as far as I know, a eukaryotic universal. Everybody does it that way. Let's take a quick look at the other end of the secretory pathway, the lysosomes, the little bags of death water. So why, first of all, are they acidic? Because they house within their membranes an ATP-driven proton pump. This pump uses the hydrolysis of ATP to power proton transport into the lysosome. So if it's pH 7.2 out here in the cytoplasm or something like that, it's a healthy cell. That's how you get to pH 4.5 in here. What's the point of that? Well, in order for lysosomes to work as the repositories of enzymes that degrade cellular constituents, or anything that cells might take up through phagocytosis or endocytosis, there have to be enzymes in there. Those enzymes are potentially very dangerous. You make those enzymes in the endoplasmic reticulum in the Golgi apparatus. Surely they're going to be chewing everything up on the way, right? Well, okay, no. Similar trick to the KDEL receptor. The trick is to make those enzymes pH sensitive, so they will only work at low pH. The various hydrolases and proteases that are in the lysosome simply don't work until they get to a sufficiently low pH. Now, lysosomes are repurposed for other things, and I also want to mention that in secretory vesicles, which, for example, neurotransmitter vesicles, might be a lot smaller, but they have that same pump making a proton gradient across the membrane, the same so-called vacuolar ATPase, V-type ATPase, is pumping protons into say, a synaptic vesicle. Those protons are then used as a power source to drive an exchanger that exchanges protons for something like glutamate, a neurotransmitter that's supposed to get loaded into the synaptic vesicle. And this is one of the ways that synaptic vesicles charge. as they're awaiting a signal to fuse with the presynaptic membrane. Furthermore, at the other end of the size spectrum, recall the diatoms, in which the cell, the volume of the cell, as we know it, is dominated by the vacuole. The plasma membrane lies right under the frustule. And there's a nucleus in there somewhere, Golgi stacks, etc. But it's sort of on an island, strung on strands of cytoplasm. And all of these vacuolar spaces here are basically just enormous expanded lysosomes. Many cells use the vacuole, this greatly expanded lysosome, as a sort of a hydraulic power system. By filling the vacuole up with sufficient concentrations of ions, some cells even use it to discard waste products and things like that. At any rate, by setting up an osmotic gradient, cells can use the vacuole as a repository of osmotic pressure to, for example, expand cells at will as they release, say, a combination of cytoskeletal and cell wall resistance to the pressure exerted by the vacuole. Now I want to look at briefly at the various fates that lead to the lysosome. Let me color code these actually. On a cell membrane, there might be events of phagocytosis that engulf large objects. And there might be endocytic events that lead to the creation of small vesicles. So for example, a bacterium might be engulfed by a phagocytic cell, and this is a kind of event in which actin-rich protrusions tend to crawl the cell membrane over an object. In contrast, in this case, in the case of endocytosis, we're talking about coat formation, say by clathrin coats, often assisted by the assembly of actin filaments that rectify this intrusion of the membrane. At any rate, endocytic vesicles fuse with endosomes. They fuse to make endosomes, fuse with Golgi-derived vesicles. Endosomes can take on topologically complicated shapes. For example, if receptors have been endocytosed, to remove them from the cell surface, then one of the strategies to keep those receptors from continuing to signal to the cytoplasm is to pinch them off into internal vesicles inside endosomes. If we look at this phagosome that's wrapped up a bacterial cell, there might be Golgi-derived vesicles fusing to build up the endosomes, of course, and also there are vesicles that recycle from the endosomes to the Golgi apparatus. That's an additional complication of no interest here to this story. The phagosome more or less immediately is targeted for fusion with lysosomes, in which the combination of low pH and strong enzymatic activity is going to degrade whatever's delivered there. So a phagosome is basically converted to a big lysosome by the fusion of small lysosomes carrying their payload of enzymes. Likewise, endosomes fuse with lysosomes. There's a third root of interest and importance, the route by which cells digest their own constituents in a process called autophagy. So if this is phagocytosis, that is cell eating, and this is endocytosis, cells just getting stuff inside, down at the bottom we'll include autophagy here in the roles of lysosomes. Autophagy is triggered when cells start to starve and have to burn their own proteins and other constituents as fuel, but it's also triggered by damaged objects that need recycling in the cell. And one that has received some attention is that mitochondria, perhaps as a consequence of their efforts, perhaps as a consequence of wearing out or something like that, as they conduct respiration, mitochondria might become dysfunctional, and components of the endomembrane system recognize organelles or other conglomerates that are targeted for autophagy. They wrap them up into double-membraned compartments, which also then fuse with lysosomes and endosomes to undergo degradation. This, if you will, is the opposite of the secretory pathway, since it takes things in from the outside world and converts them into internal materials. One of the points of interest is, of course, that the lysosome must also have pumps, channels that allow stuff, the products of digestion, that is, that allow those bits, the recycled, chewed up remains, to be transported into the cytoplasm for reuse, for example, in protein synthesis. Okay, now a final note here about the dynamics of the endomembrane system. I want you to consider the Golgi apparatus for a moment. When we talk about how proteins travel through the secretory pathway, it's easy to visualize how the Golgi apparatus is a series of processing stations in which proteins undergo successive steps in their maturation. Vesicles deliver from the endoplasmic reticulum to the cis-Golgi network, and then through a succession of transport steps through the stack to the trans-Golgi network, where they're then sent off to their final destination. But you could also view the Golgi apparatus as a successive set of maturation states for each of the cisternae, for each of the stacks. That is, the cis-Golgi network is just the first pancake in the stack, newly getting formed from the batter, that is, from all these ER-derived vesicles fusing together. And then the successive stacks represent more and more mature, more fully cooked pancakes, until the final one, the trans-Golgi network, is fully cooked and it's disassembling into vesicles bound for various destinations. This point of view on the Golgi apparatus becomes much more sensible when you think of the production of large objects. For example, some cells make collagen fibrils that might be on this scale relative to the cell. Well, those aren't going to fit into the transport vesicles very well. Some cells make ejectosomes, like the things in cryptophytes that fire off, these coiled ribbons. Those won't fit into transport vesicles very well. What about coccolith scales? Those are pretty big compared to the whole cell. Sometimes uh, they even approach the diameter of the cell that makes them. Those are products of the secretory pathway. They are assembled in the Golgi apparatus without a doubt. They aren't being transported within little vesicles from station to station. I can't even draw it on this chalkboard, but a nematocyst in a cnidarian epithelial cell might be fully half the size of the cell, even more, perhaps. And yet, it's produced within the Golgi stacks. So from the point of view of large objects, it makes more sense to consider the Golgi stack as a flux of these maturation states rather than fixed transport stations coupled by vesicle traffic. Somehow, both points of view on the Golgi must be true at once, because we can also see proteins getting loaded into vesicles and being transported back and forth between the individual membrane compartments within the Golgi apparatus, just as we can see coccoliths getting assembled within Golgi stacks and then secreted to the outside world by fusion of basically a giant membrane sac with the plasma membrane.